Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem. So today we will discuss about the CRISPR-Cas9 gene, which means the cluster regulatory interspaced short palindromic repeats. So the palindromic repeats are present in this region of the gene are present in the E. coli or some bacteria. So the bacteria contain this type of gene, CRISPR-Cas9 gene, which that is inhibit the infection of the viral DNA, which that is attached with the bacterial DNA. So the CRISPR-9 is the sequence of genes segment which inhibit the viral DNA by cutting and stop the viral genome insertion in the bacterial DNA. So here is the bacteria which contain a plasmid as well as nucleoid. Nucleoid is the double-stranded DNA which that is the present in the center of the bacteria, but without nucleus is known as the prokaryotes. So this is the prokaryotes. It's contain a DNA. DNA is the double-stranded helical structure is anti-parallel both strand which contains cytosine are always attached with the guanine, that is the nitrogenous bases, and adenine with the thymine, while that is the gene segment. The gene segment is the many repeated nitrogenous bases contain a one gene, while here is the DNA which that is also present in the eukaryotes. So this is the eukaryotes. For example, we should understand with the same diagram to understand the eukaryote. So the eukaryotes mean the true nucleus. Pro mean the before, the cryon mean nucleus. So the eukaryotes contain many chromosomes. This chromosome is the condensed formation of the DNA as well as histone protein with a coil and supercoil, and that means the condensation during the mitosis or meiosis. So this is the histone protein is wrapping around the DNA, which that is DNA is look like this, and this is the prokaryote and eukaryote DNA always contain a cytosine with guanine and adenine with thymine. So this is the gene sequence which that transcribe to produce messenger RNA, and this messenger RNA bind with the ribosome to polysome formation and translation to produce protein. While here I am explaining about the CRISPR-Cas9 gene. So here is the DNA, which that is the bacterial DNA, which contain a different region. It means the cluster regulatory interspaced short palindromic repeats, which that means it is the gene silencing of the viral DNA. So viral DNA inhibition basically not a gene silencing but the silencing of the bacterial DNA. So please remember about that. So here is a sequence which that is tracer RNA sequence and Cas9 gene sequence which contain Cas1, Cas2 and Cas3 or Cas2 and leader 1, 2, 3, 4, that is the CRISPR array which that transcribed. So let's begin to understand the tracer RNA. So here is the bacteriophage which that stimulate the leader to produce the CRISPR array, while the Cas1 and Cas2 is recognize the bacterial DNA region to produce that is with the related gene. So here the tracer RNA will be produced and Cas9 protein is produced after the translation and that's bind. Oh, anyhow, here the CRISPR repeat is also produced by the transcription. So here is the basically pre-CRISPR RNA, while here is the RNAs bind with the tracer RNA and Cas gene to combine complex, while here the RNAs bind with the this sequence like this. So this binding region is produce a CRISPR RNA which that bind, and the basically Cas gene attach with the tracer RNA to inhibit the target gene to splicing. So that means here is the bacterial DNA, viral DNA, sorry. So the viral DNA is cutting by this mechanism is known as the silencing of invading DNA via DNA breakdown. So here is look like this. This is the DNA which that is breaking down. So the recognition of PAM target sequence. So I hope you make sense about the overview. Now let's talk about the infection site, and we should here is the basically bacterial cell membrane, which that is a bilayer of phospholipid. While here is the bacterial cell membrane, and here is the cytoplasm. So the bacteriophage will land, that is a different mechanism, landing, adsorption, injection, and releasing. So the releasing after the injection of the DNA inside of the bacteria so that after the releasing that DNA will be moved inside of the cytoplasm of the bacteria. So this is the target DNA segment which that is targeting to the CRISPR Cas9 gene. So the viral DNA is here. So here is the target DNA segment. So this target DNA segment will be stimulated to produce the Cas9 gene, tracer RNA, as well as other pre-CRISPR RNA. So remember about that. Here is the target DNA. So here is the Cas1 and Cas2 is stimulate and recognize the target DNA of the viral DNA. So here is the tracer RNA and Cas9. So the tracer RNA will be stimulated to produce tracer RNA. Tracer RNA gene will be produced tracer RNA by transcription, while the Cas9 gene which that transcribe the Cas9 protein. So the Cas9 protein is also present. So it means the Cas gene will be recognized and produce the Cas9 gene. So it's binding to the tracer RNA, and combination of the tracer RNA and Cas9 protein. After the combination, on the other hand, the CRISPR array, that is the pre-CRISPR RNA, so produce from the CRISPR array 1, 2, 3, 4 segment, which that is the gene formation, and that means the transcription to produce the pre-CRISPR RNA. So this pre-CRISPR RNA will bind with the Cas9 containing tracer RNA. So this Cas9 containing tracer RNA will be bind one by one, as well as RNase 3 enzyme is bind with a different region, and that means that 3 tracer RNA Cas9 bind pre-CRISPR RNA. So this pre-CRISPR RNA will be bind with the tracer RNA Cas9 binds. So it means the basically 5' to 3' already. So it means it is the cutting side and cutting to produce the segment of the tracer RNA Cas9 binding complex. This complex is containing a guide RNA. So the tracer RNA is also known as the guide RNA, which that guides the sequence of the sequence recognition for the bacteriophage DNA. So here is that sequence of the target gene, which that is bind to cut that DNA to inactivate the function of the viral DNA. So here is the viral DNA. So remember about that. This viral DNA is attached with the Cas9 HNN domain, as well as the smaller is the Cas9 RUVC domain. So this means here is the guide RNA, which that is guide to cutting the DNA where the recognition site. So the molecular scissors are endonuclease enzyme used for cutting it. So it means here is the cutting, and Cas9 is HNN domain is the downside where the guide RNA are present, and that's viral DNA helicase enzyme used to helical and bind with the guide RNA to cut and cutting the DNA. So here is the viral DNA. So it means here is the PAM. So that means here is the non-infectable viral DNA will be produced after the formation of cutting. So it means unable to repair the cut. It means the semi repairing to inactivate that gene, and it is unable to bind with the bacterial DNA. It means the silencing or inactivate the viral DNA. While on the other hand, if we will talk about the biotechnological revolution with the understanding of the CRISPR-Cas9 gene, for example, here is the eukaryote, which that is cancerous gene will be separate after the cutting by the guide RNA, which that is the guide RNA is created by itself. So that means the new segment will be recognized. It means the new segment will be formed. So it means the we can introduce the eukaryotes. So here is the bacteria DNA, while here is the viral DNA. So the bacteria DNA is recognized, and after the recognition to it's cut. While here on the other hand, here we can also introduce the other gene via CRISPR-Cas9 for changing the gene segment for prevention of the disease. So thanks for watching. Please subscribe the channel, and if you like this video, please like.